Okay, good morning, everyone. Thanks for coming in early morning. I hope you enjoyed the dinner last night. Okay, so let me introduce myself. I am Tomomi Shimogori at Riken Center for Brain Science, CBS. Also worked as Human Frontier Research Grant Review Panel and Panel Chair from 2017 for 6 years. I was also one of the grant recipients at 2008. So the topic of the grant was to understand how thalamus and cortex get wired, what's the molecular mechanism, what the role of the neuronal activity, species differences, and also try to generate a computational modeling. So the Human Frontier Science Program is an international organization that supports frontier research in the life science. It was established in 1989 with the aim of promoting interdisciplinary and collaborative research to address complex biological questions. The organization seeks to fund high-risk, high-reward projects that have the potential to make groundbreaking discoveries in the field of life science. Let me also introduce about the history of Human Science Frontier Program. So the origins of Human Frontier Science can be traced back to 1980s. In 1987, an international committee was formed to develop the Human Frontier Science Initiative, and in 1989, the program officially launched with the first round of grant applications. The initial participating countries included the G7 nations, and the program rapidly expanded to involve more countries from around the world. The person who played a key role to establish Human Frontier Science Program was Yasuhiro Nakasone, the former Prime Minister of Japan. Nakasone's vision for international scientific collaboration and his dedication to advancing research and innovation led him to propose the creation of the program that would bring together scientists from different nations to work on fundamental questions in the life sciences. Nakasone's opinion and support were instrumental in gaining the necessary political backing and financial resource from Japan for the Human Frontier Science Program. His leadership and determination helped to transform the idea into the reality, leading to the official launch of the program in 1989. Nakasone's commitment to science and international cooperation left a lasting impact on Human Science Frontier's mission and structure. The program's emphasis on the interdisciplinary research and collaboration mirrors his vision of breaking down barriers between nations to address shared challenges through scientific discoveries. In recognition of his contributions to the advancement of science and international collaboration, the prestigious Human Frontier Science Nakasone Award has established in 2004. The award is presented annually to the individual or group who has made significant contribution to the human science HFSP's goal and ideals. Based on these histories, Japan has been recognized as a key country and supporter of the Human Science Program. The idea for an international program that would promote interdisciplinarity, research, and collaboration in the life science reasoned strongly with Japanese scientists and policymakers. Japan recognized the importance of investigating a cutting-edge research and international scientific partnership to advance knowledge and address global challenges in really early stages, which is a common sense now these days. Japan's commitment to the Human Frontier Science was reflected in its active participation in the program as a funding partner. The Japanese government and research institutions have contributed significantly to the Human Frontier's budget. Now these days, half of the Human Frontier funding is supported by METI, and the funding organization is operated by the AMED, allowing the organization to fund numerous research projects, postdoctoral fellowship, and career development awards. The Human Frontier has also fostered scientific collaboration between Japanese researchers and scientists from other countries. It has facilitated exchanges and interaction that have resulted in groundbreaking discoveries and enhanced the global reputation of Japanese life science researchers. Overall, Japan's contribution, support, and Nakasone's vision and leadership have been integrated to the success and growth of the HFSP, allowing the organization to play a crucial role in advancing frontier research in the life sciences and promoting global scientific collaborations. Human frontier grants are highly competitive And the selection process involves rigorous scientific review and assessment by international panels of experts. The program aims to fund projects with high scientific merit and transformative potential, emphasizing cross-disciplinary collaboration and innovation. And this is the panel I served for 6 years, and I obtained substantial amount of experiences, knowledge, and got understand the importance of the spirit which lies under the Human Frontier Science Program. Also made me to realize that as one of the Japanese, I'm so proud that this is the grant which we actually started. This is the grant is one, but this is the best grant, which I really want to emphasize. So as you just heard, Human Frontier is a quite unique and one and only best grant system. The application, therefore the application is also required to be very unique. So today I would like you to get a feeling what kind of experience you will get by putting the application together, and also what kind of experiences you will be able to get after you are awarded by hearing stories from 5 Japanese participants, those who obtained Human Frontier Science. They all have very unique stories behind the scene, and hope it will help you to share the spirit of Human Frontier Science and get better understanding. So I would like to invite all of the speakers up to the stages, starting with Dr. Yuzaki, who is also the co-chair of this session. So please. Okay, so once again, let me introduce Dr. Yuzaki. So Michisuke Yuzaki, he is from Keio University Graduate School of Medicine, and he was awarded on 2014. Yuzaki-sensei, please.
Okay, good morning, everybody. Thank you for coming to this session. So, oops, it doesn't work. So today I'm going to talk about 3 topics. First topic is about the advantage of the Human Frontier Science Program grant. But as Shimoguru Sensei already explained, so maybe I can skip most of the part. And secondly, probably this is something you want to hear. How can we obtain this kind of grant? And third, probably I have to give you some personal story, what we have done using this grant. So let me start with the advantage of this grant. And as Shimoguru Sensei told you, this grant really considers quirky, strange, risky, or high risk, high return type of proposal. In other words, if it is very solid, the grant is not likely to be selected. So no considerations for output indicated. I really hate outcome indicators, outcome 指標 or output 指標. And also, they don't not consider the industrial applications or patents. And I use this photo without his permission. This is a photo of famous Dr. Honjo. He often says that we don't know what will be useful from the beginning. As you know, Dr. Honjo's PD1 story started really from the very, very basic studies without knowing what can be used, what those research can lead to useful application. And I think you know them. So like Aaron Judge in 2022, sorry if you don't like baseball, but I really like. And also like Otani in this year, they are the best genius, best hitters, home run hitters. But even them, they can only hit home run in 10 to 12 at bat. So chances are very low. So in other words, if 10 grant recipients get home run type of result, that means genius, quite successful grant. So this is the point probably I would like to emphasize to AMED or granting agency. you really can't tell what will be most useful and what kind of research can lead to the most useful result at the beginning. So, and also, please forget about the 90 grantees. It's normal. And the second advantage of the Human Frontier Science Program grant is that it's highly flexible in the use. So we can only write. We are only required, asked to write a very simple annual report and also budget, very, very simple annual financial report. So you don't have to write down the detail, the usage of the grant. And third, this is probably the most important aspect, that it is really enjoyable and grant and also beneficial. So students, postdocs can have the opportunity to interact with each other, each laboratory across the continents, across countries. And indeed, in my laboratory, one of my postdocs performed experiments at Germany team, and some became a postdoc at UK team. And also international collaborative research known to have the higher chances of publishing in so-called high-profile journals. So this is enjoyable and beneficial. So then how can we get this grant, this kind of grant? So I actually obtained Human Frontier Science Program long-term fellowship in 1995, ancient time ago. And using this grant, I did my first postdoc at the Roche Institute of Molecular Biology, New Jersey, in the USA. And luckily, I could become assistant professor at St. Jude Children's Research Hospital in 1997. At the end of 1997. So because I became independent, I needed money. So I applied for the first Human Frontier Science Program research grant. I, as a leading PI, and John Connor and Dr. Fujio Murakami. And John Connor was my former boss. John Goto was a former postdoc colleague in the same laboratory. And the title was The Mechanisms of the Role of Gene Expression in Activity-Dependent Structural Synaptic Changes Using the Hippocampus. And it was not granted. And then I returned to Japan as a professor at Keio University, School of Medicine in 2003. So I again really needed to get money, grant. So I applied a second Human Frontier Science Research grant trial, and I, as a leading PI, together with Jim Morgan and Bezu in Holland. And Jim Morgan was my former collaborator, and Bezu, actually I didn't know him at all, but I just sent email and asked him to join this team. And the title was The Molecular Dissection of the Dynamics of Learning by Manipulation of a New Synapse Signaling Pathway. At that time, we already found the CBN1, and at that time, we thought CBN1 is involved in the synapse formation. Of course, we didn't know the receptors at all, but unsuccessful. And then 8 years later, we applied. I was contacted by these guys. Nobody, I didn't know anybody of them, but they just simply contacted me because they wanted to write a grant on the structure and the function of non-NMDA receptors. And because I've been studying the delta type of glutamate receptors, they contacted me. Probably they thought including Japanese researchers can be beneficial to obtain grant, but I couldn't get. And finally, in 2014, I applied. So one more time. I thought this, I usually apply the same grant until I get. So And this team started when Dr. Radu Arsesk, UK, contacted me and asked me to do some collaboration because he's a structural biology guy and trying to find a good target. And he thought parallel fiber Purkinje cell synapses and the GluD2 glutamate receptor may be a good target because they are highly expressed in the single type of synapses. So he invited me to Oxford University 2011, and we really enjoyed talking and we discussed about applying for Human Frontier Science Program together. And I met Alexander Litaev at one of the meetings. I think it's a glycobiology kind of meeting in Europe in 2013. And at the dinner time, we enjoy drinking wine and we discussed the possibility of applying for Human Frontier Science Program. So after I returned to Japan, I talked with Radu and we decided to apply one more again. And the title of this grant was The Bridge Over Troubled Synapses, a Synthetic Extracellular Protein Scaffold for Neural Activity. Probably this title may not be so good, but I will explain a little bit later. But anyway, finally, we could get this grant. So, what can you learn from my experience? First, yes, never give up. Try again, again, again. Also, I think in retrospect, I think the last time when I can get the grant, but the team members were good. So I think probably we could emphasize the collaborative elements of our grant. So, for example, I myself studying synapse biology, focusing on the cerebellar synapses, and Radu Arsesk is structural biologist, and Alexander is a behavioral scientist, and also he can do imaging, calcium imaging in the hippocampus. So it may be a good complementary combination of the different disciplines. And probably another good element is that which all members this time showed equal enthusiasm. So in the past, because I just contacted them by email, or I was just contacted by email, so we did not have the same, probably we are not on the same page. Well, simply we just wanted to have money, but the last successful application, we had the same level of enthusiasm. So all members collaboratively. I wrote the draft, but they gave us a good input. Then the question would be how you can find good members. Actually, especially for young researchers, it is often the most difficult part. And we often end up in putting your old colleagues or your old mentors, but that type of probably member will not work so well in my personal experience. Probably when you, as a reviewer, when you find your mentors or, colleagues who did the same, who did a postdoc in the same laboratories, probably the reviewer will not put the high scores, I guess. I don't know. There's no comments, but that's how I felt. So then how you can find good members? I think one of the good opportunities, just like this kind of in-face meeting, and especially when we have get-together type of parties or in front of posters. And in my case, most of the collaboration started in the get-together parties, drinking or eating something together. But the poster session, symposium, anything will be okay. But the in-face meeting is a key, not the online meeting, I think. So I think this is this kind of academic meeting is a good opportunity. Okay, and the final element is, I think it's sexy proposal, attractive proposal. So in other words, reviewers will put high scores for the innovative elements. So the proposal has to be challenging, even if it's not so much realistic. Challenging and interesting is also a key. So then I would like to explain what we really propose in my successful proposal. Shimoguri-san, so when shall I stop?
I think you hear a bell 5 minutes before.
Okay. So
still have a lot of time.
Okay, thank you. Okay, so I just copied from my application form the same figure. So it's kind of old figure. So at that time, when we applied for this 2014 regular grant, we already discovered CBN1. It's a synapse-forming molecule. It's a molecule necessary for forming and also maintaining synapses in the cerebellum. So when you knock out CBN1, the animals show ataxia. And after injection of CBN1 protein directly into the cerebellum, the same animal can now stay on the rotating rod, so that this coordination can be completely recovered. So when you look at the cerebral neural circuit, parallel fibers make synapses with the Purkinje cells. And so when you zoom up these synapses, in CBN1 knockout mice, 70 or 80% synapses are lost. So that's the reason why they show ataxia. And actually, 6 hours later, we can see the formation of synapses. So CBN1 is released from parallel fibers and connect the presynaptic element with the postsynaptic elements, neurexin and GluD2. So we wanted to make more universal synaptic connectors because GluD2 is mainly expressed in the cerebellum. So we focused on neuron pentraxin 1, which is known to bind AMPA receptors, which is ubiquitously expressed any excitatory synapses. But the NP1 is not capable of forming synapses because NP1 does not have presynaptic partners, at least in vivo. So we decided to use the neurexin binding site of CBN1 here and put it together with AMPA receptor binding site of NP1 to make a new chimeric synthetic protein called CPTX. So the idea is that the CPTX can form new excitatory synapses. So in this proposal, we proposed CPTX, and also as a backup, we proposed different synapse organizing protein, a synthetic protein, putting neurexin binding part of neurolegin 1 together with neural pentraxin. Also for inhibitory synapses, we propose that we can make nanobodies that can bind neurexin alpha and GABA receptor. It's a bispecific type of nanobody. And so using this inhibitory connector, we propose that we can make inhibitory synapses. And we also propose that by making C3, complement C3 together with neurexin binding site of CBN1, we can make this connector because C3 is recognized by the C3 receptors in microglia, and probably microglia can prune the existing synapses. And as a backup plan, we propose this kind of neurolegin 1 C3 chimeric protein. And same thing is true for inhibitory synapses. So it's really a tall story. So actually, we also did not think all of these proposal will be become successful. And also, even if we can make synthetic synaptic connector, we actually not so sure they can be useful for any good purposes. But luckily, we could make a CPTX, and it consists of CBN1 and neural pentaraction, and we showed that CPTX can form synapses not only in vitro, but also strongly in vivo. And so when we inject CPTX into ataxic model animals, CPTX can restore motor coordination. Also, when we injected CPTX in Alzheimer's disease model, 5XFAD, CPTX could restore context-aware memory, spatial memory. And finally, when we injected CPTX into the spinal cord, we could show, surprisingly, that the CPTX can restore, facilitate motor recovery in spinal cord injury models. So we could publish the paper in Science here. So in summary, I told you about the advantage of the Human Frontier Science Program grant. The main advantage is that it highly appreciates curiosity-driven science, and it's highly flexible and enjoyable. And the strategies to obtain the grant, yes, just try it again. And another key is that to find the good members, probably through in-face meetings. And we should select the sexy, appealing, interesting, risky topics. And we, based on this principle, we propose a new idea of synthetic synaptic connector, which actually worked very, very unexpectedly very well, and it turned out to be very useful for treating several disease models. Okay, I stop here. Thank you very much.
Thank you very much, Dr. Yuzaki. It's really fascinating. So we're going to have the panel session after we hear all the talks, so you can ask questions, and if there's any other things you want to know, we can answer over here. So while that way, let's move on to the second speaker, who is Dr. Jeff Wickens for OIST, Okinawa Institute of Science and Technology Graduate University. Unfortunately, he couldn't make it to be here because of the typhoon attack to Okinawa. All of the flights are canceled. Instead, he sent us the recorded video during the blackout. So let's play the video.
Good afternoon, everyone. My apologies for providing my talk as a recording, but I'm in Okinawa at the moment, and there's a typhoon outside, and I'm not sure how much battery I have left. The electricity is off, so I'll try to be brief. Let me just share my screen. I want to talk about applying for an HFSP grant, why, when, what, and with whom. So basically, today I want to talk about why to even try, when it is worth trying, what do you need for a successful application, and with whom should you apply. What I mean to do today is not tell you how to write an HFSP grant. I think that's a different topic, but to help you overcome the initial energy barrier to make an application. Just to introduce myself, I'm a professor at OIST. I've been there since 2007. Before that, I was a professor at the University of Otago in New Zealand. I've had no particular prizes and awards worth mentioning, but my qualifications today are the HFSP grants awarded in 2012, where I was a co-PI with Yale Neve, Janella Morris, and Anastasia Christakar. And then in 2019, when I was the PI with Lin Tian and Josh Goldberg. So why even try? We know on the downside that the success rate of obtaining an HFSP grant is low. And it really is a lot of work to apply. Even though there are not very many pages in the application, every word counts. So even the preliminary application, in reality, is a lot of work if you want it to be worthwhile. You have to have really good ideas, and you have to put a team together and make it work. The members of the team have to come up with their sub-projects, and it all has to fit together into one whole project. So that's a lot of work. And when the success rate is low, it might be hard to find the motivation to do that work. But on the upside, I would say that the work of preparing is very worthwhile. This is a chance to develop your best ever ideas and to get a team started that you might never work with otherwise. And the success rate is not zero. Some people do succeed in getting HFSP grants. The only way that the success rate can be zero is if you don't apply. So it's always worth applying. And it gets you into doing exciting, what we call blue sky research. That's research like, why is the sky blue? This is a little different from the kind of research that we normally have to do to get a grant. It also is structured so that you build new friendships, and it can take you in a new direction. And if you're successful, it comes with a very flexible budget that you can spend in ways that government funding may be more difficult to spend. You can use it to support your project in the best way possible, rather than to fulfill some administrative requirements. So when is it worth applying? Well, I don't think it's necessarily a good thing to apply just because you can. I think you want to apply when you really want to do the project. If you don't really want to do the project, it's very hard to make a convincing proposal. And you need to know what you want to do. That means it has to be a little bit thought through, and not just an idea that you had and put down on paper on the back of an envelope. The other thing is when you need new friends to do it with. So it's very easy, I think, to have ideas about what to do in one's own research group. But for an HFSP grant, you actually have to need other people's expertise to do the grant. And this is not the easiest thing to think through, because normally we are thinking, how can we do this? Not how can we develop a need for some collaborators? So I think it's worth applying when you really want to do it, when you know what to do, and when you're going to need some expertise that you don't have to do it. When is it not worth trying? It's not worth trying if you're not enthusiastic about the idea, because if you're not enthusiastic, no one else will be. It's not worth trying if the idea is half-baked, underdeveloped, or if it's overdone, if it's just more of the same. And it's not worth trying if you don't want to have new friends, if you don't want to get involved in working together with people in other disciplines, which can bring with it quite a few headaches and overhead. But I would say it's always worth trying to come up with a better idea. And it's probably something you should think about every year. Do you have an idea that's good to put in the HFSP proposal? So what do you need for a successful application? Well, you need an idea for a great project. And really it has to be outside of your comfort zone. If it's something that you would do anyway, it's not going to fly. You also need at least 2 other enthusiastic people who will bring something new. So it might be new technology or modeling skills, something from a different subdiscipline. And then the other thing that you need for a successful application is time. You need time to think and develop the proposal. It's not something that you can do in a week. I would say it takes a few months to prepare a decent proposal. One of the things I did with my proposal was I presented it to my research lab group, everybody in the lab group, and let them tear it apart. That was very helpful because they introduced language and selling points that I hadn't thought of. So, but that takes time. So you want to start early and give yourself time to think. You want to block out some time because you really want this to be your best idea. And then with whom should you apply? And this, I think, is perhaps one of the biggest challenges. In the full application, you have to sort of talk about hypothesis and aims, where we all know how to do that, the experimental approach.
Sorry about that. I hope it will come back soon.
Aims, we all know how to do that. The experimental approach, the novelty and significance, that's a fairly standard thing in a grant application. But when it comes to collaborative and synergistic elements in the program, you need to think about fitting together things that haven't been put together before and making them do something good. You need to write about how the collaboration originated. I'll say something about that, and how the collaboration will be conducted, which I think is a more routine thing. I think initially it's good to be a passenger or a co-PI on a grant, because you can learn a lot from what other people have done, especially if they're successful. But it can be hard to find somebody who, as a PI, wants to include you on their grant. I think there's a lot to be said for writing your own grant. As a PI, you can stitch your proposal together. You can put parts together that will make it really work. Of course, there's a challenge of writing, but maybe one of your co-authors is a good writer who can help you with that, or maybe you're a good writer. But I think there's a lot to be said for doing this as a PI. But in my case, I wrote many unsuccessful grants, and it was only as a co-PI that I had any success. And after that, I had an idea about what was good, what was going to make it fly. And so I think if you can join a team, it's very good, but it can be hard to find the team. As a PI, you have a lot more freedom and you can invite other people to the party. Often they won't come because they have their own HFSP grant going. So you might need to search for a while to find the right people. I'd just like to say a little bit more about this part of the application process, because I think it's rather different from other grants. What we came up with was a proposal that was sort of interdisciplinary, and we had somebody who could make a diagram very nicely to show how the different parts would fit together. So I think being able to put it down into some kind of organizational diagram is a very good way of thinking about how this is going to fit together. I'd just like to comment on this collaborative and synergistic elements in the program. And I've highlighted some key words in what we actually wrote. First of all, complementary expertise. So in the case of my grant, we had people working on biosensor design, others working on imaging, others working on mathematical modeling, and others working on the design of behavioral tasks. So it was very cross-disciplinary in a way, although it was drawing very heavily on things that we already knew individually. It was just putting them together in a new way. And I think this point at the bottom that this must be a synergistic effort that is going to push the boundaries of what can be measured. So in our case, biosensor design was being done extremely successfully by Lin Tian, and what we could offer was some validation and testing in vivo. With Josh Goldberg, we could bring in a mathematical model from somebody who also had expertise in imaging. And so we could put these things together. And in this respect, it's very different from other grants. Normally, you need to show a track record in the area of the grant. You need some preliminary data and maybe some prior publications. In fact, most other grants, you almost need to have done the work before you apply. In the HFSP, you can actually apply to do something that you don't know how to do. And this is not a bad thing. In fact, one of the reviewers on our proposal said that we had no background in the in vivo two-photon imaging and it would take us at least a year to get anywhere with it, but it was still funded. So I think being able to take a risk and stick your neck out, but to have friends who can provide complementary expertise and give it a little bit more legitimacy results in a very good proposal. And finally, how does the collaboration originate? In my case, the applicants hadn't met or collaborated in the past, although we knew each other's work. It happened that by chance, I sat next to one of my co-PIs at a talk which described a biosensor by the third co-PI. So we didn't know each other very well, but we knew that we were both really interested in this new sensor. Neither of us knew Lin Tian, but I contacted her cold, and she engaged in the discussion, and we had further discussion to develop a plan and collaborate on a question. We could say that the geographical spread of our groups between Japan, Israel, and Davis in the US, the high risk nature because of our inexperience and the novelty of the technology, but the high gain, the opportunity to measure things that had not been measured before, was perfect for the HFSP program. So I hope some of this is helpful. I can't talk too long because I'm on battery power and I'm liable to lose my internet connection at any time. So good luck with your proposal and thank you for your attention.
Okay, well, I hope he will get his power back very soon. Okay, well, then I think he really, outlined all what the necessity of the human frontier, and then also what the necessity of the application format. I hope you learned quite a lot from his talk. Okay, so let's move on to the next speaker, Dr. Seiji Nishizawa from Tohoku University Graduate School of Science, and he was awarded a research grant program at 2020.
Thank you, Professor. So today I'd like to talk about the novel design and the synthesis of fluorogenic molecular probes for hyaluronate. I'm now doing this work as a member of the extracellular space of the brain, a multimodal analysis from nanostructure to in vivo function. This project is supported by HFSP, and the leader of this project is Professor Valentin. And he's very handsome, and of course he majors in neuroscience. And so the other professors also major in this field, and they have a very, very good publication. But I want to say, I'm no expert of neuroscience. I'm a chemist. So you see, I'm almost alien in this team. So let me introduce my research field. I'm a chemist. I'm majoring in analytical chemistry. So I'm very interested in the design of fluorogenic probes for RNA imaging and sensing. For example, I have developed the very small molecular probes for nuclear RNA imaging. As shown here, we can clearly visualize the nucleolus by using such small molecules. And I'm also interested in triplex forming peptide nucleic acid for double-stranded RNA. This is my major field. Then, how come I joined this project? A story begins with an email from Professor Nagel. In his mail, he said, I'd like to ask you to join our team to apply for HSP. We are interested in the role of the extracellular space for brain function. So I was very surprised because I didn't know Professor Nagel. And also I didn't know anything about brain. And I also wonder why he was asking me. So his answer may be very important point. He said, we need a Japan-based chemist to synthesize molecular probes. And we are introduced to you by Professor Fukumura, Dean of your department. And then he said, your target is hyaluronan, a very interesting substrate. But I'm not interested in hyaluronan at that time. But anyway, I didn't know anything about hyaluronan, but I decided to join this program because I get the fun, I get the money, and I get to find a new research topics. So let me here introduce the hyaluronan briefly. That's a chemical structure of hyaluronan. You see, this is a polymer molecule and a kind of polysaccharide. Maybe everyone here knows the name of hyaluronan because it's a major component of beauty serum and eye drops. But at the same time, more importantly, hyaluronan is a major component of the extracellular space, and it plays a very crucial role in a variety of biological events. So here, let me confirm our research, our project interest. Our interest is the role of ECS for brain function, and my mission is to develop fluorogenic probes to visualize hyaluronan in vivo. This is my mission. This slide shows the current fluorescent probes for hyaluronan imaging. Typical way is based on hyaluronan binding protein labeled with fluorescence dyes. Yes, in this case, we usually utilize biotin and abigine interaction to label fluorescent dye. This is a common way. But very recently, fluorescent carbon dot coated with molecularly imprinted polymer named CDMIP has reported. By using this probe, hyaluronan on membrane surface can be clearly imaged as shown here. So, but in this case, in this system, the fluorescence is always on. I mean that they don't have off-on signaling ability. But in our project, we need probes that show light-up response upon binding to hyaluronan. This is a key technology we want to develop in this project. But this is very difficult. Firstly, we have to consider how to get selective binding to hyaluronan over other substrates, such as chondroitin sulfate and heparin sulfate. You see the chemical structures of these molecules is very similar to each other. So to overcome this problem, maybe we can utilize hyaluronan binding motif found in various hyaluronan binding proteins. For example, X7 peptide sequence shown here is a typical example of this. Then we attached fluorogenic dye at the end of the peptide. This is my idea to get off-on signaling ability upon hyaluronan binding. But what kind of dye should I use? This is a problem because there are no examples so far. So we need to take a trial and error approach. On the first try, I proposed the use of environmentally sensitive dyes such as DVD and thiazole orange, shown here. This is based on my research experience to design fluorogenic probes for DNA and RNA. So, and this is the actual slide to explain how to design fluorogenic probes for hyaluronan by organic synthesis. But now I think this is terrible, unreliable. Actually, unreliable. Actually, the referee report says that my field is a probe development. Obviously, they wanted to say the probe design is unreliable. In this way, we failed the first time because of me. So I felt very, very sorry for this. Next year, I came up with the idea to utilize aggregation-induced emission. TPE shown here is a typical example of this kind of dye. Very, very interestingly, TPE alone shows almost no fluorescence, but it emits very, very strong fluorescence when aggregation is formed. So we can expect this kind of binding mode. If TPE is attached at the end of the peptide sequence, this probe must show almost no fluorescence before binding. But upon binding, we can expect aggregation-induced emission because these peptide probes can bind to hyaluronan. So many peptide probes can bind to hyaluronan at the same time. Very fortunately, the referee accepted this proposal. The referee report says that I remember the application from the last year, and if I remember correctly, the problem was feasibility. But this time, they will focus on ECS visualization in vivo, which sounds more convincing. So I was very happy to hear that, and then our project has started. This is a response function of our probes for hyaluronan. You see TPE is attached here, and this is a peptide sequence. As shown here, in the absence of hyaluronan, this peptide probe shows almost no fluorescence, but in the presence of hyaluronan, the fluorescence is enhanced. This is aggregation-induced emission. And then we change the peptide sequence from X7 to P51, which has a stronger affinity for hyaluronan. We can get a more clear response. This was good. But unfortunately, the response selectivity was not so good. This is the result. You see much stronger response was obtained for chondroitin sulfate and heparin sulfate. This is because our peptide has a positive charge, so non-selective interaction is taking place between these higher charged substrates. You see the chondroitin has a minus T, the charge number is minus T, and the heparin sulfate is -4. So non-selective interaction is taking place with these substrates. We were very disappointed with these results, but anyway, we need to develop as a type of fluorogenic probes. Then I focused on FRET, fluorescence resonance energy transfer. Actually, this is a very common way to design fluorogenic probes in biochemical research. So in this system, we utilize two kinds of dyes. One is a donor and the other is acceptor. When two dyes are close to each other, fluorescence is quenched due to energy transfer from donor to acceptor molecules. But two dyes get away, the fluorescence is recovered. So based on this idea, we attached FITC donor here and the double acceptor is here. Then we expect this kind of binding mode. Before binding, the fluorescence is quenched, but upon binding to hyaluronan signal, fluorescence is recovered. But to be honest, this is just expectation, because we are not sure how the structure of peptide changes upon binding, but we didn't have a choice. We decided to take a chance. Very fortunately, this system works very well. As shown here, the fluorescence is reduced in the absence of hyaluronan, but in the presence of hyaluronan, the fluorescence is recovered. And also, interestingly, the response selectivity became better as compared to our previous system. You see the fluorescence response, chondroitin sulfate and heparin sulfate is almost same to that of hyaluronan. I'm not sure for this reason, but anyway, we can get a very clear fluorescent response for hyaluronan and the selectivity became better. So we applied this system, this probe to live cell imaging. This is a result. So so hyaluronan concentration is elevated in variety of cancers. So here we analyzed HeLa cell. You can see very clear fluorescence here on cell membrane surface. And this image is very similar to that obtained by CDMIP. So we expected maybe we can visualize hyaluronan in living cells. This was confirmed by co-staining experiments. Here we utilize hyaluronan binding proteins labeled with fluorescence dye. You can see clear fluorescence here by using our FRET probe, and at the same time, at the same place, we can also see very clear fluorescence by using hyaluronan binding protein. So you see the fluorescence of our FRET probe is well overlapped with that of hyaluronan binding protein. Now I can say this probe is actually binding to hyaluronan without off-on signaling ability. Thanks to this signaling ability, we can visualize hyaluronan without any washing steps. This is a wash-free imaging of living cell with FRET probe. You can see very clear fluorescence here, along the cell. So this is a significant advantage compared to conventional system. Okay, let me summarize my talk. Today, I'd like to talk about the design and the synthesis of fluorogenic molecular probes for hyaluronan. Actually, to my best knowledge, this is the first probe with off-on signaling functions that can image hyaluronan in living cells. So, and finally, I'd like to thank Professor Nagel for choosing me as a Japan-based chemist, because, there are so many chemists in Japan. And also, I'd like to thank Professor Fukumura for introducing me. Otherwise, I couldn't join this exciting project. Okay, that's all. Thank you for your kind attention.
Thank you very much, Dr. Niso. That's a beautiful story. Okay, so let's move on to the next speaker, Dr. Azusa Kamikochi from Nagoya University Graduate School of Science. And she was awarded as the Early Career Grant at 2011 and also the Research Career Grant in 2021.
Thank you for introduction, and it's my great pleasure to share some of my experiences with Human Frontier Science program. Okay, so the goal of my research is to understand the animal communications. So I'm very interested in how they can communicate to each other, how they can understand their communication signals. So these are examples of human, birds, elephants, and ants. So they are doing something, communicating. So my specific question is how their brains understand these communication signals. To tackle the question, so we are using two model animals. The first one is the fruit fly. So during the mating behavior, the male fly sings to the female to attract the female. So he vibrates his wing to generate a very unique type of sound. and which is known as courtship song. The song has a species-specific temporal pattern or song rhythm, and the females with their brain, female evaluates the song rhythm to decide if she accepts the male or not. The other animal, model animal I use is mosquitoes. So for the mating purpose, they form this swarm. And in the swarm, male detect female very tiny wing beat sound. And we like to understand the neural mechanism underlying these acoustic communications. So these are the experiences with my experiences, personal experiences with Human Frontier Science Program. So I have actually two experiences. First one is the Young Investigators Award, so between 2011 and 13. So let me start with this first experience. The title was From Genes to Circuits, the Evolution of Species-Specific Communication in Drosophila. So we are from UK, Japan, and USA, and Belgium. And the purpose was to elucidate the evolutionary, material, and mechanistic basis of species recognition in the fruit fly acoustic communication systems. And the team members consist of specialists in biomechanics, neuroanatomy, bioinformatics, and neurophysiology. So my contribution was, as a Japanese scientist, I use my expertise in neuroanatomy to contribute the project. So let me share how the project started. So 2 years before we submitted a proposal to the Human Frontier, so it was in 2009. So I attended a Cold Spring Harbor meeting for neurobiology of Drosophila in the United States. And after this meeting, I traveled to some of the fly labs in the States, such as Iowa and Caltech. And at Caltech, I met Mara. So she's now a professor of neuroscience in Princeton University. So she just found me and talked to me her intention to apply a Human Frontier Science program with me. And to be honest, at that time, I didn't know what Human Frontier Science project was. But anyway, it sounded interesting, so I just jumped in. And the topic was about acoustic communication using fruit flies. So I recruited Dr. York Albert. He is a friend of mine, and his expertise is on the topic. So I recruited him, and York then recruited Dr. Stein Art. Stein is a bioinformatics specialist. So at the beginning, in 2009, directly after I came back to Japan, we started a discussion. But at the beginning, we didn't know much well with each other, and we are in different countries, different time zones. So discussion is based on Skype online communication system. So it was totally new experience. To me, everything was new to me. For example, we are living in various time zones, so we have to carefully choose the appropriate time for discussion online. And the discussion styles are so different between countries, especially Japan and USA was very different for me. And it was challenging, of course, for me to write a proposal in English because I have never experienced that. But anyway, thanks to the strong leadership of other members, we managed to submit a letter of intent, which is used for the first screening, so which is actually a very simple one. It's not so difficult. And successfully we passed the first screening, and then submitted a second one, the complete version of the proposal, which was a very detailed and long one. It was very hard actually for me, but anyway, the hard work paid off, and we successfully got the award. So, and then at the first round, the York, he's a professor at University College London, UK now. I'm moving to Germany now, but he reached the first team. So he was a PI, principal investigator of the first of my Human Frontier Science project awarded. And actually he was a colleague of mine when I was in Germany as a postdoc. So we know each other very well, actually, and he's still a good friend of mine. And a decade after this first round of the project, we happened to start a second project, a new project, together again on mosquitoes. So the second one was started, the planning of the second one was started in 2017. So York invited me to a Royal Society meeting in UK, so known as Theo Murphy Conference, organized by the Royal Society. So it was a good chance for me to visit his lab for the first time. So after the conference, I visited his lab in London. So his lab is located in the Ear Institute, UCL, in the heart of London City. So it was a very nice building, and so he had a lab there. So in the first Human Frontier Science project, we worked together on fruit flies. But after that, York has started something new. So he started mosquito researches after the first project. And we talked about potential future collaborations there, and to make that happen, I talked with everyone in his lab, and then I finally found Dr. Matthew Su, and he's now an assistant professor in my lab now in Nagoya. And Matthew there at that time, and he expressed a keen interest in starting mosquito project in Japan, Nagoya. So I was lucky to find him there in 2017. And 2 years later, we finally managed to have them have a mosquito team in Nagoya University. So this is Matthew. This is Matthew, and the other is our first student, Yumin. So we set up a new mosquito room and measurement setup. And this shows how we measure the flight tone of mosquitoes. And we also can now measure the electrophysiology response and calcium imaging and behavior, many types of behaviors. So we would like to understand the neural mechanism of their mating behaviors and auditory systems. So as you may be aware, the mosquitoes is the most dangerous animals for humans. They kill a large number of people every year due to the viruses they transmit, like dengue, malaria, West Nile fever, and Zika fevers are transmitted by mosquitoes and killing humans every year. So we are focusing on these two important disease vectors, the Aedes aegypti and Aedes albopictus. The Aegypti mosquitoes are domestic in South Africa and other tropical regions like South Asia and Africa. And the albopictus are domestic, as you know, in Japan in summer this season, and of course other subtropical regions. So these two mosquito species bite humans and transmit dengue and Zika viruses. So these are both disease vectors. So, but from a biological perspective, the mating behaviors of mosquitoes are highly interesting. So they have the mating behavior in mid-air, so they form swarms, large groups of mosquitoes for mating purposes. In the swarm, mostly these are males, and sometimes a female just flying, passing by. At that time, male has to detect the female by female's flight tone, and approach toward the female by hearing the flight tone of the female. The response is very robust and known as phonotaxis. So after male find a female by sound, he approaches. So actually, many of the mosquitoes approach to a single female, and one lucky guy can cooperate with the female. And after mating, female start blood feeding to obtain nutrients for egg laying. So the aim of our study is to understand the process of the mating behavior to explore the possibility for intervention to control mosquito populations. So we started discussion again online. So the primary platform for this round was Zoom. And we have again team members from different countries. So Japan and UK, again York and Doris in the United States of America, and Dr. Chen in Taiwan. Okay, so this is the outline of the mosquito project. The biophysics and the modeling aspects are covered by UCLA and UCL. And these components are closely linked to the lab experiments in the lab performed by Nagoya University, our team, and UCL. And additionally, we have experiments in the semi-field condition organized by Dr. Chen at National Health Research Institute in Taiwan. So let me share the outcomes so far. So at the first round, so the aim was we explored the evolution of species-specific communication in Drosophila. But to be honest, it was a very big challenge for 3 years period. So I published the first paper on this project in 2013, but the second one has to wait till this year. So I should confess that the goal we set was indeed very ambitious. But the experience of the first round has its beneficial aspects. So the experience of having that, organizing the first round, helped me a lot to, it serves me as a stepping stone for initiating the second round. So with that experience, we can have now more realistic but still challenging plan. And we improved our communications between team members for collaboration and having a managing task to help facilitate the collaboration and coordination. Thanks to that, the second one so far goes better, and we already published first paper last year. So in my opinion, the significance of Human Frontier Science project is that so it allows a researcher a chance to tackle challenging project. And even if the results doesn't go well and it doesn't match with your initial expectations, it will help you for another chance. And being a part of team members allows valuable knowledge exchange among international members. So personally, it helps me, it plays a crucial role in helping me to pursue my ultimate goal that is to decode animal communications. Okay, I'd like to acknowledge the team members of the first round, Mara Yorkstein, and the second round, York Dolores and Junhong Chen, and especially thanks to Dr. Mashipol Su to organize the second round project. Thank you for your attention.
Thank you very much. It was a really nice example of a very successful story. Okay, so then let's move on to the last speaker, Dr. Ikue Mori from Nagoya University Graduate School of Science.
Good morning, everyone. So I will talk about other for fifth member or something. Anyway, I have experience in Human Frontier Science program in an old system that I just wanted to emphasize at this point. And first, I want to talk briefly about what I'm doing using C. elegans. So I'm working on a behavior called thermotaxis of C. elegans. When the C. elegans animals are cultivated at a certain temperature, which is ranging from, say, 15 to 25 degree with ad-lib food, which is bacteria, for, say, maybe 3 or 4 hours. So this is conditioning. And after that, those animals are placed on a temperature gradient of this temperature ranging from like 17 to 23 degree, and left for 1 hour. So animal, it would be like 100 to 200 adult animals. And what we can see at the lower panel, that the distribution of the animals migrating toward the cultivation temperature, which is 17 on the left, and 20 at the middle, and 23 on the right. So I thought, I came back from US, which I received a PhD. Directly, I got a position as assistant professor at Kyushu University. I started on this project, completely changed from my PhD study. So that the thermotaxis, I thought, is very interesting because behavioral plasticity can be directly observed at the behavioral level, which is depending on the past cultivation temperature. So I got associate professor position after being assistant professor at Kyushu University for 9 years. In 1998, I established my own lab as associate professor. So this is a picture of 2001, like 3 years after establishment of my lab. A staff member is just only me and 2 postdoc fellows and the students who seek to receive the PhD at the end anyway. So this is a picture when Corey Bergman, now at the Rockefeller, previously UCSF, visited our lab. So we are so young. Anyway, and then I just wanted to emphasize that about 2010, I decided to comprehensively study the C. elegans behavior. I thought I need an approach which is very much interdisciplinary, so that we can develop the sort of microscopic systems for calcium imaging, of the genetics, and auto tracking of the moving animals, and etc. other techniques. So I recruited as a staff member, for example, like a computer scientist and a theoretical physicist. So those the members together, like recent staff members. We in the same lab we can do computation and molecular biology and genetics as well. So I make a big decision myself, because if I expanded the lab to be very much interdisciplinary, I could not go back to a very small lab. So but anyway, I got Human Frontier before I came up with this idea. So this is a neural circuits for thermotoxic behavior. The very sort of you can using the taking advantage. You can take advantage of C. elegans as a model, like a systems level approaches you can do. So temperature is received by individual sensory neurons called AFD and AWC. And we knew that AFD also is a memory temperature neuron. That we knew that. But anyway, in the downstream, we have like interneuron circuits, which detect innate state of the animals. So that the temperature received outside by sensory neurons sort of integrated with the innate state, which was as information stored in interneurons downstream. And then finally, the neural computation will generate behavioral outcome using like a muscle, body or muscles. So that's the sort of the view at least my lab would come up with. And that was the C. elegans neuroscientist will come up with in a similar way. For example, like I just wanted to emphasize that the one postdoc, Takuma Sugi, found that the systemic temperature sensing system is working in C. elegans through HSF, heat shock factor. So under this regulation by HSF1, we found like estrogen. Estrogen is released from the muscle cells, for example, to influence AFD temperature reception, for example. So those like cell-cell communications are very, very important. And recently, the Moon-Soon Jan in the lab is conducting to answer that cell-cell or tissue-tissue interaction mechanisms. She recently found like a glycosylation in stem cells. Stem cell is like a stem cell for skin. Stem cells for generating hypodermis, for example. So so GLY12. This is a gene for glycosyl transferase. This particular GLY12 gene is functioning, expressing in a stem cell, not in a nervous system. But if we knock out the GLY12, you can find defectiveness in the behavior. So the nervous system is affected by non-nervous cell tissues or cells. And glycosylation is also very important that the Moon-Soon Jan found recently. So that's sort of a brief introduction of myself and my lab's study. And finally, the goal of my science would be how environmental information in sensory systems are integrated with innate and stochastic activity in inter and or motor neuron circuits, thereby generating coordinated behavior. Because C. elegans have a sort of stereotyped patterns of behavioral elements. So I just wanted to talk about my experience in Human Frontier Science Program. So just to emphasize again, I applied in all Human Frontier systems anyway, from 1991 to 2001. We tried 6 times, and so my success rate is 50% because the 3 times we were awarded. So first one was we was awarded in 1991 with 5 members, 3 from Canada, 1 from US, and 1 from Japan, which is me. And for your reference, the age of applicants are 30 to 50 years old, and I was 34 at that time. So as my scientific career, I was a assistant professor in Kyushu University. And after this sort of support for 3 years, in different members, like 5 members, we applied, and the proposal was rejected. And the next year, 1995, the proposal was rejected. The 5 members were different from the members that we applied for in 1994. And finally, we are again, second time we are awarded. So in this case, we thought about the one from Netherlands and one from US and one from Japan. And so I moved to the Nara University at this time during this awarded period. That was very nice. And 1999, the 5 members, including myself, rejected again. And in 2001, we means like one from UK, one from Italy, and one from USA, and one from Japan sent application, and we were awarded. So that was very nice. Like I was all the members are before about 40 years old, all the members. And I just wanted to emphasize the whole Human Frontier, all system teams, that was probably similar to the new system now. Team must consist of scientists from different countries. The aim of the grant must conduct innovative basic research. The category of grants was research grants only. We didn't have a young career, whatever grants. The review process of the proposal was just one step. We just applied, and we knew whether it was awarded or not. And the success rate in general was higher, I think. So probably say over 10%, 10 to 15%, as opposed to maybe about 5% in the current system. So from my experience, in the old system, what I found, asked by AMED to give a talk about the Human Frontier Science Program, and when awarded, all members, 3 to 4 members, are individually very strong in their area by accumulated publications. And again, everybody said, awardees are said, we set the research goals to solve fundamental questions of no compromise in neuroscience. I think that's very important. No compromise, no complication and compromisation. And research plans, because we can sort of C. elegans neuroscience field, a lot of technologies are going into in say like early 2000 or something. So research plan included technological advances of no compromise that were available at the time of application. That's when we were awarded. I think that was for sure. And again, we designed a very innovative and challenging project that will be pioneering the field. I think that's very important, pioneer the field. And I want to emphasize this sentences. All members, team members are thought partners, sometimes not collaborator, and no competitor, of course, and very much importantly, trustworthy colleagues, devoid of any scooping unpublished results. So we sit down and say about, what we are, so like very from our heart, we are sort of connected as a scientist and respectful to each other. I think that this is very much, very important. Thought partner, no competitor, no scooping, but all. So we are very relaxing and enjoying the conversation with people in the team and to make up a proposal. So maybe this is my idea, maybe biased, but when we didn't succeed in getting grant was when we want to have some new field. So we want to have recruited one scientist, which is relatively young, for example. So that just finished a postdoc in abroad, for example, in the US, and came back to, for example, Japan to establish a lab. Sometimes he or she proposed very unfocused and I can do everything stuff. And those like 1 out of 4 members, for example, have this kind of very unfocused and too much challenging, some passion, didn't really work, in my opinion. And also, one time I remember, sometimes the members have to compromise, regretfully, to include the professor when the young scientist asked to do so. So I regretted to do that. So as I said, that all the members in a team has to be equally important with each other and enthusiastic to the project with each other. So there is no up and down of the enthusiasm among the team members. That's, I think, very important. And this is the last slide. I owe Human Frontier Science Program my sincere thanks for supporting my tenure of scientific career from assistant professor to associate professor period. Lessons learned: the international team must consist of trustworthy scientific colleagues, not merely a place to develop a friendship. We have to scientifically evaluate. I think this is also important, but although it's kind of very difficult to say in front of the audience, but we have to scientifically evaluate the level of both colleagues and ourselves to try to predict the future scientific direction. And I think this is important because the success rate is so low in this program. So our mission as awardee of a Human Frontier Science Program is to push the frontiers of basic science forward. So we have to be aware we are in that kind of position. I think that's important. So that's I just wanted to say. Thank you very much.
Thank you very much, Dr. Murray. That's also another good example. And as you have seen that, she's one of the frequent visitors to Human Frontier. So here we still have some time. So I want to ask all the speakers to come up to the stage and probably we can have a panel discussion. Sorry, Dr. Murray, I should have let you know to not go down. Okay, then, well, once again, thank you very much for all speakers for a really nice talk. And I hope that you guys got a lot of, good ideas about what Human Frontier is and what kind of experience you can get from applying the grant and also after getting the grant. But here, also the symposium is meant to, encourage a lot of you guys to apply to the Human Frontier grant. If you have any questions regarding how to apply, or also any question to all the participants here today, for those we heard the talk about, please do come up to the microphone and feel free to ask whatever the question you have. but I was ready. These kind of things, it's very difficult to encourage people to ask. So one other thing, which I want to ask everyone is like, well, you guys have already shown us about how you found your partners. But now these days, when I talk to people, they often say like, it's very difficult to find a partner. And if there's any suggestion from you guys, how to find good, team member, I would like to hear about that. Do you mind if I ask from Yuzaki sensei?
As I showed in my slide, in most cases, I met these people at the in-person type of academic meetings, like not the SFN, a little bit smaller. I think this meeting is okay. I mean, annual meeting of Japan Neuroscience Society is okay, but there are several good academic meetings, and especially, when you attend a meeting, of course, the presentation and listening to the presentation is okay, but I think the most important thing is to, interact with each other, get together type of parties or in the break sessions. But anyway, in my case, all my collaborations started like that way. And sometimes I'm directly contacted by email, but even so, without meeting in person, as I showed you my failure record, we could not share the same level of enthusiasm, and maybe we could not, as Mori-sensei said, we couldn't trust completely with each other, so those kind of applications did not work out very well.
I see. Yeah, I think we have good examples here. For instance, like, now we have emails and also we have Zoom, so we can actually interact with each other. And then because, the Human Frontier proposal is supposed to be novel collaboration. So if you already have publication before, you have to declare that this proposal is completely different from your previous record, right? So in that case, we are highly encouraging people to team up new people, which means that you probably don't know each other. And Dr. Nishizawa, you actually have a really good example. You were suddenly contacted by those people. Can you tell us a little bit more about that story?
I think my case is very, very rare. So I just accepted his offer, but I hesitated to join that program because I didn't know anything about neuroscience, and he had Ronan also. But I thought it was a very, very good chance for me to join such an international program. So that's why I decided to join that program. So don't be afraid of missing such a chance. Be sure to get such a chance is very important, I think.
I'm glad that the email you received didn't go into spam mail. Right.
Question to you. So why Dr. Valentin Nigel thought it is important to include chemists, Japan-based chemists? I think it's a great thing, for the development of Japanese science field. So, any idea?
Maybe he likes Japan actually, so he has developed a very high resolution spectroscopy microscopy named sushi. Sushi, actually he likes sushi. So actually for his system, he needs a new chemical tools to emphasize the resolution. So that's why he needs some chemist to synthesize chemical molecules. Yeah.
So it was based on the scientific necessity and the personal interest.
Yeah.
Well, actually, that's a really good point, because when we think about, when we are postdoc, when we think about where to go, we actually think like, well, the science is really important, which lab you want to go, at the same time, which country you want to go is also one of the things. So if you want to go to a new place, it might be good to find your collaborator from those countries, right? And one thing about having Japanese in the member, we had a rumor before, people were saying you probably have better chance to get the grant, but that was actually just a rumor. Because we don't review, because whether you have Japanese in a team or not. But one thing is, when we say this is supposed to be the international collaboration, we also consider intercontinentality. So in that case, if you say okay, we have 4 different European countries, we think it's a little bit Europe oriented. And in that case, it's really good to have someone from different geography, like Japan is actually a really good place. So that's also not because Japanese, but geographically, it is actually really beneficial to have Japanese in a team. Okay, so the other is Kamiko says is probably a really good example, because she already had an example for having it from a very early career, and then probably that was a surprise for you to be join in. But based on that, you also had a second round, which actually made you to shift the field. And based on your experience, how much human frontier work? I mean, did human frontier push you to go there, and then how much it really pushed you to go farther or expand your research?
Yeah, so it was just because I met York there in London, and I just wanted to start something new, and Human Frontier Science Project is a very good platform to start something new without any publication related on it. So actually, I probably didn't have any chance to apply Kakenhi for starting something new with no record. But Human Frontiers, they didn't matter. So if it's challenging and if it's promising, and they would take it. So it was a good platform for me to start this project. So it pushed me a lot to start something new.
That's really great. So as we heard a lot from them, is Human Frontier don't ask for preliminary data when you apply for the grant, and we also don't even ask what's your budget plan is, because we really don't know what's going to happen. So if it is basically, we just judge, whether you have a really, ambitious goal and whether the team member can make it to happen. So this is the most important question. And I think Kamiko Sensei is a really good example to do that, which also made her to push her field, because other key word when we review the grant is whether they are leaving their comfort zone. So if it is an extension of your ongoing work, we won't take it. So we want to see the challenge. We want to see the people who's pushing their limit. And I think she's a really good example for that. And one good thing is a human frontier, as we have already seen. Once you get it, people understand about the human frontier. So we have people who got it multiple times, and Mori Sensei is the best example for that. Mori Sensei, can you tell us about what you think about human frontier, since you've been so successful person?
Well, actually, that really helped because, as I said, I shifted from Drosophila actually to C. elegans when I was a PhD student in the US, and I didn't know any Japanese when I came back to Japan. So it was kind of I had a lot of colleagues and friends in international countries rather than in Japan. So that was, to me, as a human frontier, was probably the best and very natural grants that I can apply. So that was very nice. And so during the 3 awarded periods, the Human Frontier supported me for 10 years as a junior kind of career to become before becoming a professor. So it was very nice, and we can sort of because we know emails and the Skypes at the time, and we knew each other, and that was I don't know how to say, it's very nice and very natural having that money.
Yeah, it's a really nice story. Okay, after hearing everyone's talk, I think there's someone who wants to ask a question.
Yes, yes, I have a question about the composition of the team. So do you think it's important to have at least one senior PI, or would it be okay to team up among only junior PIs? Any ideas?
Sure. Who wants to start? I mean, well, Mori Sensei had the age range and group.
Actually, yeah, when one time we were, so our team was unsuccessful, when I think the new, like a young scientist established a lab in Japan or something. So he or she was so enthusiastic. But in like a sort of culture, I don't know, Japanese culture, so he or she wanted to have the professor in the lab. And we couldn't resist. We couldn't say no. And we were failed. So I would, from my experience, you can go ahead to have a team with only junior researchers. I think that's no problem. I don't think, yes.
I think there are two categories now, and Kamiko-san got the first
young investigator hours, but actually the competition rate is not so, almost the same between the program grant and young investigator hours. So it doesn't matter if you apply either of the project.
Probably I can put a little bit of follow-up information as a reviewer side point of view. And why we do have these two categories is because Human Frontier asks for new collaboration and also asks to, make yourself, leave your comfort zone. But for the early career people, like, someone who just got the lab, how you can leave your comfort zone, right? So when we review people who they're only, young investigators, we don't be so harsh about how much they leave the comfort zone. But at the same time, you can just team up with like old people. In that case, you better apply to the research grant in that case, even you're in an early career type of thing. So it depends on the, proposal and how much you're leaving from your comfort zone. But like, Kamikochi Sensei said, like, we are generous about those, but the rate of the success rate doesn't change among those two. So we just basically care about the pure science, how good it is.
Okay, thank you very much.
Okay, so also for that, from the reviewer point of view, I also like to add, like, we don't look at the age range, actually. Yeah, we don't evaluate people because you're younger, you're old, or whatever. We just don't care. Like, again, we just care about the science. And what is also interesting is we don't look at genders. This is really interesting. Now these days, we should care about it, right? But again, we care about science, and it comes later. But at least the review panel is very international, and gender balance is really amazing. Yeah. So we are all balanced team that review in a very fair way. Okay, so that was a good question. If there's any other question from the whole, please come up to the microphone. And if not, we can actually continue discussing about the grant application. So as again, like, it's very difficult to team up. That's one big hurdle. And also, like, what we heard from Jeff is actually, it really takes a long time to put the proposal together. So I was wondering how long it take for all of you guys to put the proposal on the full proposal, not LOI. Well, it can be from LOI, but the full proposal takes a really long time. And like, how often you had to meet? You ever had any disagreement with your team member? How you solve that problem? If you have any story that you can tell us about, I would like to hear about it. Please.
So in the current system, first you have to write
Letter of intent?
Yeah, letter of intent. I was about to say COI, but no, letter of intent. So if you are selected, you can go to the second stage. And Actually, I became PI in the US and had to write the RO1 grant, and compared with RO1 grant, it is very small amount of effort for me, and it's more like a Japanese grant to me. So, so in my case, I always write the first draft in most cases. It took 1 month or so, but the last 1 week, it's really amazing. We communicate each other and add something. But in total, it doesn't take so much. So, but you simply have to write what is the innovative part and what is the collaboration part of those kind of stuff. That's something we Japanese are not so far familiar with. Not the science research plan itself. Research plan is not so difficult because we always write grant application, kakenhi or RO. But in human frontier science program, I think we have to spend we have to write many times. What is the collaborative part? What are the interdisciplinary or what are the innovative aspect of this grant? So, and you really have to convince the reviewer. That's a little bit difficult for us. And then you can get the good support, advice from other team members.
But that's also the part you should have a good discussion with your team member. Otherwise, you won't be able to come up how to write down those collaborative elements, right? And Hisawa-sensei, probably, because you come from very different field, you probably had to spend a long time talking with your team members.
So yes, actually yes. So in my case, the leader, Professor Nagel, organized everything, and he wrote almost all story. And then the other members put some piece to complete his story. So in my case, I am a chemist, so my mission is very clear. My mission is to develop the chemical probes for hyaluronate and something. So anyway, the leader wrote to make a whole story, and the other professor put some piece to complete his story.
That's good. But probably they actually, had to really think about how to integrate to each other. So it can't be like, one person generate a probe, one person generate a technique, one person's going to use it. It's like a one-way collaboration. And we immediately reject that kind of proposal. So it should have a really good integration to the both direction. And probably it already had that kind of element. I mean, it was clear, right? They had to have this kind of probe to do whatever they wanted to do. But I'm sure they had an opposite direction as well. So this is something you want to remember when you're going to put the proposal to apply for the human frontier. And what actually I'm surprised about Kamikochi-sensei's proposal is because you guys already had grant before, right? And then this is the second round. How did you convince the reviewer?
Please ask the reviewer for that. But
well, to be honest, I think I reviewed your proposal because I was there.
I don't know. So, but it's that new project, a new model animal. But actually, the topic is similar with the previous one. I don't know why you took us. To be honest, yeah, it was maybe good.
Yeah, so this is the thing. Even if this is not the novel collaboration, if it is going to bring them to the new direction, this is something we really like. And then it actually had a really strong component. It's just not just a reprint of the fly system that you had. It was going to take you to the completely different direction. So we were completely convinced about that. Yeah. So once again, so this is the reason why we don't write in the, how to apply. It can't be the, old collaboration or anything like that. It's still, even this is the, old collaboration. If there's a synergy happen here and they're going to bring them to the new direction, we definitely going to take it. So that's the reason why we don't write it. It's prohibited to have a old collaboration or anything like that. But just you have to remember, it's going to take you to the new direction. Right? So, okay, so finally, Mori Sensei, who's, again, I'm so curious about, because you've been so successful, probably you're the one who's most successful among the human frontier. But what do you think? Do you want to encourage people to keep applying? Because you've been someone like that, right? You never gave up. So how you could keep that motivation?
Like maybe Kamikochi-san's case, I have one colleague from America. So actually Corey Bergman and I, plus Italian from UK or something. And we are just enjoying, Corey and I are really enjoying. So we kept, we applied 6 times. And 3 times we failed. I mean, Corey failed in Human Frontier.
Well, this is the reason why it's from Human Frontier. We don't care who you are.
Yes. But it's, I don't know, we just want, of course the project is different, but C elegans anyway, and behavior in general. But that we are taking like a challenging technologies and the methodologies, and to go into the different directions in every proposals. And we just enjoyed, as I said, no scooping results, unpublished results. And we are really enjoying the conversation, and so as to develop a new project. That was really fun part. I couldn't stop doing it. I also I was asked by some other scientist whether I can sort of join to their team, and I refused to go. We are kept applying, so for 10 years. That was really fun. So I would encourage people here to, anyway, you just challenge. Because like 90% of the people are rejected anyway.
Probably Yuzaki Sensei have that experience, right?
but it doesn't really matter.
Yeah.
To make it improve, that process is very important. For you to be, for us to be like a sort of matured in science. I think that's it.
Yeah, so since Yuzaki Sensei as well, like try so many times, like get your grant application rejected is not a good feeling. But did you ever get any feedback after like a full application? I mean, LOI we can't give that much feedback, but what about in case you were rejected after the full application?
Unlike R01 grant, we cannot get good feedback from Human Frontier. So we cannot tell why we failed. So actually, in that aspect, it would be nicer if Human Frontier Science Program Reviewing Committee can return some comments back. But in my case, I had no idea.
I see.
That's simply I stick to it.
So if it is really far from the border, we probably don't give feedback. But there's some people who actually reapply next year. In those cases, who is really close to the border, we actually give them some feedback. Yeah.
That means we are not close to the border.
Probably so. But probably if you ask them, they can actually give you some feedback, because we really discuss a lot and we take a lot of notes. So we definitely have a lot of information. And we're running out of time, but for the review panel, like what we do in the end is we really spend a long time to discuss about one proposal. So the reason why I remember your proposal, because this is how much we had to read everyone's proposal. And this is actually really nice. You review this paper and then see really beautiful outcomes is happening. This is a really nice experience to be a reviewer. And this is something we can actually do as a Human Frontier. So I hope, everyone got an idea what this grant is, why we are doing this. And at the same time, being as a Japanese, this is a really great system, which actually happened more than 30 30 years ago. It was a really good insight how, international collaboration is necessary, how you can open up to the new field. And this has been so successful. And if you just go back and look at the list of the awardees, there's so many Nobel laureates who actually already received the human frontier and been very successful. So I hope you got a good idea, and then we could encourage everyone to apply to the human frontier proposal. I'm looking forward to see your proposal one day. And thank you very much, everyone, to be here today. Thank you.